Base pairs keep the double strand of DNA together. If we think of DNA as a twisted ladder, each rung is a pair of nitrogenous bases, such as adenine and thymine, or guanine and cytosine. Remember that there are 4 bases in DNA, and their sequence spells out the information DNA carries. You can think of genes as the words or sentences they spell. The pairs are stuck together, not with real covalent bonds, but with a weak attraction we call a hydrogen bond. Each nitrogenous base has a partner. In DNA, adenine and thymine pair up, and so do guanine and cytosine. In the related molecule RNA, thymine is replaced by its close relative, uracil, so the pairs are adenine-uracil and guanine-cytosine. The base's ability to pair up comes from their nitrogen atoms. They are called nitrogenous bases, after all. Each nitrogen, shown as a letter N, has a pair of electrons that makes it slightly negative, so it can be attracted to the slightly positive hydrogen atoms on its partner, shown as a letter H. We call this a hydrogen bond, and it's drawn as a dashed line in the illustrations. Each base has a place where it attaches to the sugar phosphate backbone of DNA, or RNA. In our ladder analogy, the 2 backbones are the 2 vertical parts of the ladder. The hydrogen bonds occur between the base on one strand and its partner on the other strand. As you can see in the illustration, guanine-cytosine pairs are connected by 3 hydrogen bonds, and adenine-thymine pairs are connected by 2. Adenine-uracil bonds, not shown, are also connected by 2. The attraction we call a hydrogen bond isn't a real chemical bond. It's like when a sock sticks to your pants after you take them out of the dryer. You can easily take the sock off, it's not permanently attached. But an electrical attraction keeps the sock there temporarily. Since the hydrogen bonds are so weak, they can be easily broken when it's time to make a copy of DNA. Enzymes break the bonds, unzipping the double helix into 2 separate strands. Because each base pairs with a certain partner, the enzymes that build the new daughter strand can easily read the old parent strand and use it as a guide. For example, if the parent strand has an A, the daughter strand needs a T to match up with it. This is the key feature that allows DNA to be replicated. RNA doesn't always exist in a double strand. Sometimes it's just a single strand, and that means it can fold back on itself. The bases pair to each other, creating hairpin shapes, as shown here. This specially shaped RNA can then act like an enzyme to do other important jobs in the cell. The illustration here shows a transfer RNA, which helps to build proteins. Base pairs occur when nitrogenous bases make hydrogen bonds with each other. Each base has a specific partner, guanine with cytosine, adenine with thymine in DNA, or adenine with uracil in RNA. The hydrogen bonds are weak, allowing the DNA to unzip. This lets enzymes replicate the DNA. In addition to keeping DNA together in a double strand, another function of base pairing is to allow RNA to take on complex shapes, which lets it do other jobs in the cell, such as building proteins.